Have you ever wondered why sour flavors seem so distinct and sharp compared to other tastes? What makes our tongues capable of detecting that zesty tang of lemon or the sharp bite of vinegar? The answer lies in a fascinating interplay between biology, chemistry, and neuroscience, one that scientists are only beginning to fully understand. But here's where it gets even more intriguing. Recent research has uncovered a key player in this process that not only helps transmit sour taste signals, but also keeps the cells responsible for detecting sourness alive. This discovery could reshape how we think about sensory biology altogether. Our tongues are remarkable organs, brimming with tiny structures called taste buds that allow us to experience the rich tapestry of flavors necessary for both survival and pleasure. Each taste bud contains specialized cells designed to pick up specific types of tastes. For instance, type 2 cells are responsible for sensing sweet, umami, and bitter flavors through receptor-based channels. On the flip side, type 3 cells specialize in detecting sourness, a task they accomplish using mechanisms reminiscent of those found in neurons, including synaptic vesicle release. Yet despite recognizing these differences for years, researchers have struggled to pinpoint exactly what drives this synaptic activity in sour-detecting cells. Enter a team of dedicated scientists from Okayama University in Japan, led by Professor Ryusuke Yoshida and Assistant Professor Kengo Hori. Their groundbreaking study, published on June 24, 2025, in the Journal of Physiology, reveals a critical piece of the puzzle. The protein, SNAP25, plays a dual role in sour taste perception. Known for its importance in facilitating neurotransmitter release in neurons via the snare complex, SNAP25 was previously thought to operate exclusively within the nervous system. However, this new work shows that it doesn't stop there. It also supports the long-term survival of sour-sensing type 3 taste cells. This finding bridges two seemingly unrelated functions, neural signaling and cellular health. To better understand SNAP25's role, the researchers created genetically modified mice lacking the SNAP25 gene, specifically in their taste cells. These knockout mice appeared normal and lived to adulthood, but something striking happened inside their taste buds. The population of type 3 cells, the ones responsible for detecting sourness, was drastically reduced in two major areas of the tongue: the fungiform papillae near the tip, and the circumvallate papillae further back. Meanwhile, type 2 cells associated with other tastes remained unaffected. This stark contrast highlighted just how crucial SNAP25 is for maintaining sour-sensing cells. Digging deeper, the team used a technique called EDU tracing to track the life cycle of type 3 cells over time. They discovered that while new sour-sensing cells continued to form at normal rates, they couldn't survive beyond about 2 weeks without SNAP25. This wasn't due to problems with cell creation. Instead, the issue lay in sustaining these cells once they'd developed. This revelation pointed to an entirely new survival mechanism tied directly to the function of synaptic proteins like SNAP25. Electrophysiological tests provided further confirmation of SNAP25's importance. By measuring electrical activity in the chorda tympani nerve, which carries taste information from the front of the tongue, the researchers observed significantly weakened responses to sour stimuli, like hydrochloric acid and citric acid, in the knockout mice. Interestingly, reactions to other tastes such as sweet, salty, bitter, and umami were unaffected since those rely primarily on type 2 cells. This clear distinction reinforced the idea that SNAP25-dependent synaptic transmission is uniquely vital for processing sour flavors. Behavioral experiments added another layer to the story. Using a test known as the breath access licking paradigm, which quantifies how much animals avoid certain tastes, the researchers found that wild-type mice strongly avoided sour solutions. In contrast, mice missing SNAP25 showed much less aversion. While this indicated some residual ability to detect acidity, it suggested the existence of additional systems outside the typical gustatory pathway. To explore this possibility, the team created double knockout mice lacking both SNAP25 and type 3 cells and TRPV1, a proton-sensitive ion channel involved in somatosensory detection of sourness in trigeminal neurons. The results were eye-opening. Double knockout mice exhibited almost no behavioral response to sour liquids, confirming a dual-system model for acid detection, one involving SNAP25-mediated synapses in type 3 cells, and another relying on TRPV1-expressing neurons. Even more surprising, extremely high concentrations of acids still elicited weak responses in these mice, hinting at yet unknown backup mechanisms or redundancies in sour taste perception. Such safeguards likely evolved to protect against potentially harmful acidic substances. At its core, this study transforms our understanding of taste bud physiology. It demonstrates that SNAP25, a protein traditionally viewed as exclusive to neurons, is indispensable not only for transmitting sour taste signals, but also for ensuring the structural integrity and longevity of sour-sensing cells. This dual functionality underscores a broader principle. Synaptic proteins can serve multiple roles, linking signal transduction with cell maintenance in sensory tissues. Professor Yoshida summed it up eloquently. SNAP25 isn't merely a facilitator of neurotransmission. It's essential for the very persistence of sour-sensing cells. Without it, these specialized cells vanish, erasing our ability to perceive sourness. This insight challenges conventional wisdom about synaptic proteins, suggesting that sensory epithelial cells actively participate in neural communication and self-preservation. Beyond pure science, the implications are profound. Understanding the unique nature of sour taste signaling, this research paints a vivid picture of how molecular machinery and sensory biology intertwine, revealing that taste buds use neuron-like vesicular release not only to convey sourness, but also to sustain cell viability over time. Such insights enrich fields ranging from neuroscience to cell biology, emphasizing the complexity of sensory epithelial tissues as interfaces critical for life-sustaining functions. Looking ahead, the team at Okayama University continues to push boundaries, combining molecular genetics, electrophysiology, and behavioral science to uncover how taste receptors influence feeding behavior and energy balance. With over 70 publications under his belt, Professor Yoshida's group remains at the cutting edge of taste signal transduction research, shedding light on the intricate dialogue between sensory input and systemic physiology. As our grasp of taste bud architecture and function grows, so too do opportunities for advancing public health, nutrition, and food sciences. Insights into how SNAP25 orchestrates sour taste transduction and cell survival may eventually pave the way for improved diagnostics or treatments for taste-related issues, enhancing quality of life for countless individuals. So, what do you think? Should we reconsider the roles of synaptic proteins beyond traditional neural contexts? Could there be other hidden players in sensory biology waiting to be discovered? Share your thoughts in the comments below. We'd love to hear from you.